All right, thanks everyone. Welcome to Frontiers in Psychiatric Treatment. This is our second installation covering depression with James Murrough, MD, PhD. I'm Kat Morris. I'm the Communications and Marketing Director for the Department of Psychiatry at Mount Sinai. Just to give you a little introduction to Dr. Murrough, he is Associate Professor of Psychiatry and Neuroscience at the Icahn School of Medicine at Mount Sinai. And he's also the director of the Depression and Anxiety Center for Discovery and Treatment. And as part of that center, he also directs the clinic, which is the treatment-resistant depression program. And with that, I'm just going to pass it over to Dr. Murrough to cover depression for this webinar. Thank you so much.
Thank you, Kat, and great to be here. Okay, let me share my slides. Okay, well, it's really a pleasure doing this, and thank you to everyone who's attending virtually. I'm really excited to present this. It was a little bit of a challenge. There's so many exciting things going on right now in depression, and I'm really going to try to limit this to 45 minutes to leave time for discussion at the end. So let's jump right in. Oh, sorry. A couple quick disclosures. I do a little bit of consulting for a few companies. I am named on a patent for actually one of the treatment areas we're going to talk about today about KCNQ channels. It's a filed, not awarded. There's no royalties. We're also going to be talking about ketamine and esketamine quite a bit today. And although I don't receive any money, I'm not named on it. There is a patent held by the school and the dean around the use of ketamine and esketamine for depression. So that's an important disclosure for everyone to be aware of. And this is more about that, what we call an institutional conflict around ketamine for depression. So like I say, an ambitious plan we have today. A very brief background on depression, because that's not the point, but I'm going to touch on a few things about the presentation of depression, about its neurobiology very briefly, really to sort of set the stage for some of the new treatment approaches that we'll be talking about. And with any luck, I'm going to highlight new treatments that focus on GABA and glutamate, inflammation, opioid systems, KCNQ channels, psychedelic, and neurostimulation. So if we think we can do it, let's get started. Okay, depression is by some measures could be called the world's largest health problem. This is because it carries so much disability. It affects young people. It's common. And for, unfortunately, many people, it is recurring and progressive. And it's the #1 cause of suicide in the United States. And as you'll see, and as you know, although many of our treatments are highly effective, unfortunately, there's a significant minority, maybe about a third of individuals that really don't respond well to our current treatments. And that's really why we're here today. By latest counts, about almost 5% of the world's population will have a depressive episode during any 12-month period. That's an astounding number. Most depression episodes do remit within 12 months, but on the other hand, recurrence is quite common, and it's up to north of 80% in clinical care settings. It's not the focus of the talk today, but I would be remiss without mentioning suicide. It is the second leading cause of death among young individuals. And as I noted before, depression is the most common psychiatric disorder among individuals who die by suicide. So the thought is, if we can make inroads into how we can effectively treat depression, hopefully that'll be part of bending that curve on the sort of alarming actually increase in suicide. In terms of how depression presents, as you all know, it's highly heterogeneous. This is one diagram about this. It has all these, it's basically a constellation of symptoms. But one reason I want to show this is because, and I'll give some examples of this later in the talk, some of the thinking is these systems, whether it's fatigue, amotivation, cognitive problems, they have their own brain systems, their own treatment targets, and we might be better off really honing in on a group of a more narrow symptom dimension rather than trying to just treat depression as a whole. To that end, I just wanted to, many of you are aware of this probably, this is what's called the RDoC approach. This was put out many years ago now by NIMH, but it's meant to be sort of an orthogonal, maybe not an alternative, but an orthogonal approach to think about how to classify individuals from a research perspective to not consider just the DSM diagnosis, but these different domains. And then each domain could be measured across multiple levels of analysis. Again, it's sort of to encourage a different way of thinking. And I'll show you some examples of clinical trials that we've done and others that have tried to take this approach. I promised to have something very short on neurobiology so we can focus mostly on treatment. This is a nice, very high level schematic from a really large tour de force Lancet review, actually that Helen Mayberg pointed me towards, who was also an author on this that just came out in Lancet. I would encourage you to check it out. But basically at a systems level, we know that there are mood regulation systems that involve limbic emotion generation systems, and then prefrontal and cognitive control systems, and that somehow the interaction of these systems has gone awry in depression. And so, you tend to have sort of dominance or overactivation of some of these areas that are indicated in black, like ventral cingulate, amygdala, and conversely, a relative reduced functioning or underactivation, sort of some of the cognitive control regions like prefrontal cortex. So that's sort of a high level sort of neural systems view of what might be going on. A note on inflammation, because I'll come back to this as an example of some of the emerging treatment approaches. So if you look in the middle of the slide, you see a brain and you see some of the circuits that we just talked about. And going back to an earlier slide, the thought that we can maybe map some specific symptoms of depression onto these. So perhaps reduced response to pleasure, anhedonia, we can map to sort of basal ganglia and ventral prefrontal, whereas things like anxiety, which is an important driver of depression in some individuals, may be more linked to things like dorsal ACC and amygdala. And what the author is showing here is that there's various ways for dysregulated immune signaling to impact these circuits. And so the thought is, the changes in the chemistry affect the circuits, which then gives rise to the specific symptoms. So, for example, in the upper left, it does seem to be the case that patients with depression, on average, have small but measurable increased circulating pro-inflammatory cytokines, and these can impact these circuits in different ways. So there's two ways are being shown here, or actually three different ways that's actually relevant to some of the approaches we'll be talking about later. One is that they can deplete synaptic availability of some of the monoamines, and that's shown in the upper left. It also might influence glutamate metabolism through different mechanisms in a way that would be maladaptive. And finally, so some pro-inflammatory pathways actually reduce BDNF dependent signaling and reduce neuroplasticity through those ways. And these are all ways in which depression could be caused or exacerbated and could be targeted for future treatments. I think this is my last biology slide, and I think, again, this is some people are splitters and some are lumpers, but this is sort of a heuristic. This is a review from the late great Ron Duman from 2016, but the idea of a cellular basis of depression as sort of loss of plasticity, loss of synaptic function in regions like medial frontal cortex and hippocampus seems to hang together. In rodent stress models, you have loss of these dendritic arborizations, which is shown on the left. And again, we know that some of the important players like the monoamine transmitters, cytokines and other things impact this pathway. And in fact, chronic treatment with standard antidepressants can reverse this atrophy. And then if we'll see things like rapidly acting interventions like ketamine can do this quickly. So, this will be coming back to this model of sort of treatments that can reverse sort of stress-dependent loss in neuroplasticity in synaptic function, sort of maybe a final common pathway, if you will. So let's get to treatment. So let's start with the good news. There's many evidence-based, highly effective treatments for depression. We know this is a good thing. There are many forms of evidence-based psychotherapy, pharmacologic treatments, and neurostimulation. As I alluded to before, there's some limitations. If we zoom into a classic monoamine synapse cartoon, we know this is where our current first-line antidepressants work by increasing the availability of monoamines in the synapse, ultimately having downstream effects in postsynaptic neurons, not all of which are still amazingly completely understood, but do converge on pro-plasticity cascades, most likely. In terms of how we're doing, again, there's a lot of ways to look at this, but this is a reasonable way. This is a review from a number of years ago now, but I think it makes the point. This is simply showing response rates, so the proportion of individuals that have a good antidepressant response. If we look at the cluster on the right, just focusing on major depression, that's 165 studies, north of 44,000 patients. On average, you can see that response to antidepressants is significantly better than placebo, about 55% response to drug versus 38% of placebo, but it's 55%. And we've seen that number, we could look at STAR-D. We know as you get more treatment resistant, it goes down, but these are actually from RCTs of FDA registration trials. So it's likely that in the community, these numbers are a bit less, but we're somewhere between 50 and 60% effective, all comers, getting someone out of their episode within a reasonable 8 to 12 week period with current treatments. So obviously there's room to do better. Let me spend the next few minutes on ketamine. So I had a chance in my own training and early career to be involved in this research at Mount Sinai. There's a schematic from Charney and Manji on the left in Science, kind of recapitulating what we've talked about. So even then, the theory was if you can make out the figure, they're showing that, ultimately standard monoamine antidepressants are likely impacting on second messenger systems that ultimately increase cellular plasticity and resilience, in part through BDNF-dependent mechanisms. And in the green, if you can see, this is the authors describing theories in which something targeting glutamate and the NMDA receptor, ketamine's an NMDA receptor antagonist, could sort of rapidly lead to that similar effect. And that still seems to be sort of holding true. Ketamine there were a lot of firsts. So it seemed to work quickly. And I'll show you some of the early studies on that. Seemed to be effective in some patients that were so-called treatment resistant. They didn't do well. There was also an early signal that it had anti-suicidal effects. And overall, this was in the middle to 2000, between 2005, 2010, 2015. We saw a lot of effort in academia and in the pharmaceutical industry suddenly focused, turning away from classic monoamine and serotonin to focus on the glutamate system for depression. I'm not going to spend time on this. NMDA is an important glutamate receptor. It's an ionotropic receptor. Glutamate, of course, is the primary excitatory neurotransmitter in the brain. Importantly, it gates calcium. It has a key role in neuroplasticity, and classically, it's been implicated in learning and memory. Ketamine, as you see in that figure, so it occupies the channels, and it's considered a high affinity channel blocker, as are some other drugs. 3 early studies on racemic intravenous ketamine. This is an anesthetic that was looked at, repurposed, off-label. These early studies used a single dose, a low dose of 0.5 milligrams per kilogram. That's about a quarter of the anesthetic dose. So the first study is on the left. That's Berman. That was about 8 patients. And what you can see, that top panel, is change in Hamilton depression score. The time scale, which is along the bottom left, is on the order of minutes. And that first mark after the double hash is 24 hours. So you can see the line. You give the injection, ketamine is rapidly metabolized. It's out of the body in a few hours. And within the next few days, interestingly, in this study, even 72 hours after the single dose, they were continuing to feel better, continuing to improve. So strange, unusual. By the way, ketamine caused altered mental state, dissociation. That's what's shown in the bottom 2 left panels. A feeling of high, and even a quasi-psychotic like could be paranoia. This study really came about because ketamine was being used at Yale as a model of psychosis in healthy volunteers. In any event, the study of whether ketamine had rapid antidepressant effects after a single dose was followed up by Zarate in 2006. You see a similar curve. This was the same dose, intravenous, 1 injection. And what you can see is a large separation on the Hamilton 1 day after a single treatment. And then we replicated and extended that work. That was published in the American Journal in 2013. That's on the bottom right. Here, the main difference was it was a larger study. It was a 2-site study funded by NIMH. And here we were giving ketamine versus a different anesthetic. The other studies had been ketamine versus saline, and there was concern about the blind. So here we put ketamine up against another anesthetic, and ketamine, again, at 1 day after a single infusion, that was the primary outcome, showed a superior antidepressant effect. So this was sort of the early studies. Fast forward, skipping over many, many studies, 2 years ago, 2+ years ago now, maybe be 3 in March, the FDA approved a form of ketamine, Esketamine, in a nasal formulation as the first of a new class of antidepressants. This screen grab is actually from the FDA panel. So again, don't have time to get into the details, but I would encourage you to take a look at it. On the left just shows an example from one of the positive studies, which led to the FDA decision. In these studies, adults with treatment-resistant depression were started on a new oral antidepressant plus a saline nasal spray or esketamine. And you see the 28-day treatment period with improvement, a change from baseline in the depression scores in both groups, but with some superiority for the esketamine. And the more detailed results from the different trials that went into the FDA approval are shown in a table from the New England Journal of Medicine paper on the right. Again, if there's time in the comments, we can have more of a discussion. There was some debate, not all the studies were positive, but in any event, it gained approval. Interestingly, and we won't be getting into it today, esketamine subsequently received approval for, I think, the first ever FDA approval for what they called major depressive disorder with suicidal thinking, maybe a year or so ago. So I want to spend a couple of minutes actually. So we do administer esketamine as part of our treatment clinic at Sinai. So I want to get into the weeds a little bit. This is all right from the package insert, but just some things in terms of the safety and the dosing. So first on, safety is on the left, dosing is on the right. So safety-wise, a few important contraindications, which are rare but can happen. And this is all really about the main risks of ketamine, which are: 1, a transient, but in a small proportion of patients, a significant increase in blood pressure. So if you have a history of aneurysmal disease or other problems, AV malformation, that would be a contraindication for that patient. In addition to elevated blood pressure, it causes dissociation, cognitive impairment. Individuals can't drive for 24 hours after the treatment, and there may be toxicity to the fetus. The most common reactions are dizziness, dissociation, nausea. Typically the way we do this, so this esketamine was approved as part of what's called a REMS program. So it's very tightly controlled. A clinic or treatment center has to enroll in the program to be approved to get the drug through special pharmacy. Patients have to be approved by the REMS program to start. They come to the clinic twice a week for 4 weeks. We measure vital signs, get rating scales. They then self-dose with a nasal applicator, remain in a private treatment room, and for 2 hours at the site after treatment. And then if they feel steady, they can go home. So that's very briefly how the treatment works. The induction phase, as I mentioned, is twice a week over 4 weeks. And then the idea is that then we make a decision as to the response or not. If there is a response, then the labeling would suggest a maintenance phase of weekly for a few weeks, and then every other week or less frequently, with the goal similar to ECT, as once you can get an acute response, you then want to reduce the frequency as much as possible, with the goal being to prevent a relapse and giving as few treatments as you need for a relapse. So it really becomes sort of very tailored to the patient at that point. Some of the what I didn't mention in detail is, well before esketamine, the use of off-label IV ketamine became relatively prevalent, at least in the Northeast, California, maybe all over the country. Not regulated by the FDA, hard to get data on it. but Santa Cora published probably the best guidelines and suggestions on how to use recommendations for off-label racemic ketamine in 2017. We updated that and included Esketamine in an American Journal review that was just published last year. So this is just a snapshot of that. But for more details, I would point you to that, suggestions about how to, again, patient selection, monitoring for either the IV racemic ketamine or esketamine. I did want to include on the bottom there just a note on route of administration. So for the esketamine nasal spray, that's, you know, what it is. But for the off-label Racemic, classically it's been studied and administered IV, but it's worth noting it's available by almost every route of administration you can think of. Again, there's not much in the way of sort of randomized controlled studies on most of this outside of IV. But you can find case series, anecdotal information on things like intramuscular, which seems to have almost so-called 90% bioavailability, similar recommended dose, 0.5 milligrams per kilogram. Intranasal, we published a study years ago, Lapidus et al. In Biological Psychiatry, where we gave a single 50 milligram racemic intranasal dose. I haven't seen much published since then on racemic intranasal. And there's been some reports of oral being used in the community, sometimes multiple times a day at the doses I've indicated there. But again, that typically has very low bioavailability, but there's not much in the way of data on that approach, but that is being done. So before I move away from glutamate, this is a review we published a few years ago now, but it makes the point. So I've given you some examples, and really I think part of what's been exciting about the ketamine experience is that it has encouraged vigorous research in this area. So this is just a cartoon of a glutamatergic neuron. The presynaptic neuron is at the top, the postsynaptic at the bottom. As you now know, ketamine's an NMD receptor antagonist, but what I've done is each of these points where I've indicated with a number, these are all different ways that the system could be modulated, and in fact, where there's active translational or clinical research programs actually doing this, that one way or another by all these different methods could ultimately increase, again, BDNF dependent proplasticity and synaptogenesis, which ultimately we're sort of viewing as the, you know, final common pathway. So let me turn now to the glutamates, the other side of the coin, which is GABA, okay? This is the most common inhibitory transmitter in the brain. And another first for the FDA is the drug Brexanolone is the first drug approved for the indication of postpartum depression. This was approved just a few years ago, around when esketamine was similar. Brexanolone is a formulation of allopregnanolone, which is a neuroactive steroid, a derivative of cholesterol, and happens to be a GABA A receptor positive modulator. So it doesn't act like a benzodiazepine, but it acts at a different site as a positive modulator. And this was a unique drug that was developed as a single treatment, a single dose, given IV over 60 hours. And what you see here is some data from one of the two of the phase 2 studies, I believe, in Lancet from a few years ago. That was part of the FDA package that ultimately led to its approval. Also with a REMS program, it seemed to be very well tolerated, but there were some cases of severe sedation or loss of consciousness. So there's a REMS program in place there. It's been unclear what the uptake in the community of this drug has been. I believe it's been challenging to figure out how to get it to patients, to postpartum women that are in the postpartum period. They develop depression, they're at home with baby, they would need to come in somewhere for 60 hours for an infusion. It's I think been a little bit of a problem, but in any event, at least in terms of a mechanism, and maybe there'll be future formulations down the road that can make this simpler to administer, but it was an important discovery and an approval. And moving to the next slide, I think kind of to that point. So again, this is just one study. This happened to be in the New England Journal of Medicine a couple of years ago, a small phase 2 study. There's been several studies since then. Same company, Now this is an orally available synthetic neurosteroid, similar to allopregnanolone in its mechanism of action, as I understand it. Also a GABA A positive modulator. This is SAGE-217. This study is in adults with depression. And what I'm showing you here is just an example of one of the studies. This is a 15-day daily dosing regimen. And the authors are reporting a very large benefit of drug over placebo in a 90-person study with a very large between group difference of about 7 points on the HAM-D. So this was, again, obviously very exciting, novel GABA modulator. There was subsequently a negative phase 3 study, and I checked their website and looked like earlier this month, they announced a positive phase 3 study. So it's not entirely clear where this stands, but this is an active area of investigation, and this may be, medicines like this may be some of the newer ones we end up seeing becoming available sooner rather than later. So a little bit of a delay on the slides. Okay, sorry about that. So let me wrap this up before I move to the other another area I want to talk about. Apologies for the busy slide. There's probably not much you need to take home from this, but again, I'm trying to keep some themes here. So now this is also from Ron Duman in Molecular Psychiatry 2019, now trying to tie together the ketamine NMDA rapid antidepressant story in terms of its effect on cortical synapses with these observations of Brexanolone, SAGE-217, GABA modulators that you see indicated on the lower left. Again, trying to fit this in one sort of heuristic understanding that in both of these cases, they may be impinging on similar downstream mechanisms to reverse the maladaptive synaptic effects of stress. Okay, so let me turn now to inflammation and a couple other topics we wanted to hit. If you remember back to that list. Very good, very recent review in Nature Reviews Drug Discovery by Drevitt. So that kind of has everything you need to know to be up to speed on targeting inflammation for depression. What I'm showing you on the left from that article is that, you know, some of the key pro-inflammatory cytokine targets that we think might be targeting depression. You see TNF alpha, I think I already mentioned that, and infliximab. There's been some monoclonal antibodies targeting IL-6 and others. But here's the quick summary that I tried to put on the right-hand side of the screen, and then I think I have a little bit of data to show you on this. So there are consistent meta-analytic support for elevated pro-inflammatory cytokines in depression, but they're small. And overall, the results of anti-inflammatory interventions in depression have been mixed, I would say. There have been studies even of COX, non-specific or COX-2 inhibitors. Some have been positive. Some have some methodological issues. The most exciting studies where a specific pro-inflammatory factor like TNF alpha or IL-6 could be targeted, those have been negative. And those are the two that I'm aware of that have randomized controlled data in depression. So not in patients with immune disorders that have depression. I'll show you a little bit on that. But in patients with depression that don't have an otherwise underlying immune disorder, these trials have been negative. Now, they've been negative with some important caveats that I'll show you. For example, for the TNF, anti-TNF infliximab trial, it was negative, but if you looked at patients with high CRP, C-reactive protein, post hoc, it benefited them. If you look at IL-6, it was negative on the general depression score, but actually it was positive on a secondary outcome of anhedonia, which kind of goes back to what we were talking about earlier, the target. So there's been some, I would say, some cautious optimism, some very interesting data with minocycline, which is an anti-inflammatory antibiotic So I would say, again, watch this space. It's ongoing. This is also from that review I mentioned. The depicted on the left is a partial table, and it's what I was just describing. So 2 trials of TNF alpha, not significantly different on the primary, but some interesting secondary analyses. A fairly well-powered IL-6 study, which was disappointing. It was negative. That's in that 3rd row. That has not yet been published. However, it was superior on the SHAPS, which is an anhedonia scale. So like I say, some reason for optimism, but too early to tell. On the right, what you see is a set of meta-analyses. Now, this is looking at antidepressant effects in patients with immunological disorders that are being treated in RCTs for these. And there, if they happen to have comorbid depression, there's a pretty strong signal in some studies also controlling for improvement in general immune or medical symptoms, that there is an antidepressant signal in those patients. So that's kind of what we know now. Let me spend the next couple slides. Let me transition to talking about the opioid system. And I really want to focus on these next 2 slides just on the Kappa opioid receptor. So very briefly, there are multiple opioid receptors. The one that we all mostly know is the mu receptor. That's the MOR here on the slide. The mu receptor is responsible for its analgesic effects, euphoria, and presumably for its abuse liability of mu agonists, morphine, all those. Kappa, so stress activates Kappa. Preclinical work shows pretty consistently Kappa signaling leads to dysphoria, anhedonic behavior in rodents. So the idea is blocking Kappa. So don't touch mu, block Kappa as an antidepressant, anti-anhedonic strategy could be promising. So that's what I wanted to bring up here. And I've just outlined a few examples of this. So naltrexone is a non-specific, non-selective mu agonist, but also Kappa antagonist, although it is mu preferring. There's been just a few small studies on actually naltrexone in depression. Buprenorphine is a partial mu agonist, but it's also Kappa antagonist. And again, a few studies suggest benefit at very low doses, actually, in TRD. I would say the bottom 2, and I have another slide on the J&J compound, the bottom 2 are probably the most active. So the Alkermes compound is a combination of buprenorphine and samidorphan, which is a mu blocker. So you put the 2 together and you isolate the Kappa antagonism effect. And so there's been several studies in depression on this specific compound. They're mixed. That's all the time we have to get into. Some were positive, but I would say the development of that is uncertain. The FDA asked for more information. I'm uncertain if the sponsor is continuing, but there were some promising signals there. So I think that idea is still in play. Let me tell you a little more detail about the J&J compound. So this is the only selective Kappa antagonist, Kappa blocker that I'm aware of. We had the good fortune to be involved in this large multi-site study led by Andrew Crystal that was published last year in Nature Medicine. So this used that very same Kappa antagonist, and this was sort of an example of what the clinical trial of the future might look like. So here, the primary endpoint was brain. Okay, it was a functional MRI of reward response in the nucleus accumbens, right, in the striatum, using a drug that should target this area based on lots of detailed preclinical work that I'm not showing here. This was an 8-week trial, and it enriched. And to get into the trial, you had to have anhedonia. It wasn't focused on a DSM diagnosis. You had to have some mood or anxiety. So we had bipolar, PTSD, GAD in here. I think a lot of us were worried what it was a very unusual study, not huge, 89 adults, And in some ways, it was very successful. So what I'm showing you on the top left is the primary response. So treatment with drug over placebo increased reward response in the brain as hypothesized. On the left, that's the SHAPS, our old friend, the SHAPS score. This is a self-report of anhedonia. So lower scores are better in terms of response to pleasure. And here, the drug had a nice separation from placebo. So this was quite encouraging and sort of, I think, makes a couple of the points I'm trying to make in this talk about neuroscience-based approaches and possibly more of an RDoC approach. Let me tell you about some of our own work that's similar in the sense that it's focused on anhedonia. It takes sort of a translational neuroscience approach. So again, we're focused here on the nucleus accumbens VTA reward circuit. And there's a drug, retigabine or ezogabine, that is an activator of a channel that, long story short, in preclinical studies, by targeting this channel, it normalizes the function of the circuit it wrote models, and it's antidepressant. And we chose ezogabine because we could repurpose it because it's a drug that's approved for seizure disorder. So under the auspices of another NIMH experimental medicine protocol, this was just published last year by our group, Costi et al. In the American Journal. Similar approach, same brain target, same functional MRI approach. This was a 2-site study with Sanjay Matthew at Baylor. So there you see the hypothesis again is the treatment with ezogabine over placebo should increase brain response to reward. That's what you see on the left. It didn't quite meet statistical significance, but the effect size was actually the same as in Crystal. The table on the bottom left shows all the secondary outcomes. These are all the clinical measures. So you see MADRS, QID, SHAPS. There's a lot of numbers there, but if you look at the column with the p-value, it's fairly striking. These were all highly significantly improved under drug versus placebo. And I'm just showing you a couple examples on the right-hand side of the figure, the graph. MADRS is in the top right, and then SHAPS on the bottom right. We're conducting a second follow-up study now using this same approach. So let me rapidly turn to psychedelics for depression. Large topic, but a couple of things I wanted to point out. There was a nice review from 2020 in American Journal. If you look at the table on the left, you have sort of what might be considered, classic psychedelics. So this would be things like psilocybin, LSD, DMT. These are really serotonergic antidepressants, thought to really have their primary mechanism of action by binding and activating the serotonin 2A receptor, among others. And then you have things like MDMA, we're not going to talk about today. That's in a very successful development program with PTSD. And then interestingly, ketamine comes back. And so sort of not a psychedelic exactly, but it's sort of in this space. It causes altered mental status and dissociation and might have some psychedelic-like effects. Now, on the cellular level, if you look at the bottom left, this is just a little bit from, again, just trying to make a point here, from Cell Reports in 2018, where the authors conducted a series of studies showing that, again, classic psychedelics seem to increase spine density, neurite growth, synaptogenesis, in a way that they argue is similar to chronic treatment with antidepressants or ketamine. So there you see on the left, treatment with vehicle, the control, and then DMT, which is a classic psychedelic, and ketamine in terms of number of spines. So again, I'm trying to give you guys a coherent concept here. And likewise, showing that these psychedelics worked through some of these same TrkB dependent mTOR pathways that have been studied as a mechanism of ketamine for depression for many years now. Still have a little delay here. Okay. All right, so let me show you one bit of data. Okay, let me focus in on psilocybin. I think that's where there's been the most work in depression. And there's been, I think, just 2 studies. Both were published in the last, say, 2 years. The first in JAMA Psychiatry, you see on the left. This was 2 sessions of psilocybin in adults with depression. They were randomized to a treatment versus a delay. So it's not a prospective RCT. Well, it is, but the control condition is simply a waiting list. In any event, they found a substantial and robust antidepressant effect, as you see on the left, in the immediate treatment. That's the orange squares versus delayed treatment. And these individuals had two on weeks 2 and 3. They had a psilocybin session with therapy that lasted about, I think 11 hours. You can see it's a small study, only 13 and 11. This was the first RCT I'm aware of for psilocybin for the treatment of depression, very small study. On the right, study that's gotten a lot of attention in the New England Journal of Medicine last year, a little bit larger. I'm sorry, I don't have the sample size on there. I think it's more like high 40s per group, not huge. And here, this was an interesting design. Individuals got also a 2-day dosing schedule of psilocybin, a similar dosing range to the Davis study. But this time, individuals were randomized to that or a first-line SSRI, escitalopram. And the primary outcome was change in the QIDS, depression. And what you see on the top panel, top right, there was actually numeric superiority for psilocybin, no difference statistically from escitalopram. So you could look at that as it wasn't better, but then again, escitalopram isn't bad. So there's sort of some conflicting discussions about how to interpret this. But in any event, the primary outcome for this study was no difference between the interventions. But then I think a lot of people were excited about some of the secondary outcomes. And without getting into a lot of the details, this was just one that was in the main figure of the paper on the bottom right. That's an indication, that's a wellness measure where you got much higher scores under psilocybin with wellness. So very exciting, much more work to do in that space. In my last few minutes, so I do want to save time for questions, I want to briefly touch on neurostimulation. I want to touch on DBS, VNS, and TMS. Obviously, can only say a few words about each thing, each being a large topic in and of themselves. But thank you to Dr. Mayberg for this slide. Helen directs the SEACT program at Mount Sinai, one of the leading DBS programs in the world. Her work, some of it is depicted here. Again, this, if you look on the left, this is very neuroscience-based approach where, again, there's sort of a dominance of an overactivation of some of these ventral emotion regions interacting with some more cortical regions. But the initial finding was really in depression, abnormal activation of the subgenual cingulate in PET imaging. That's what's shown in the middle panel. And so the idea was to target that. And over the last number of years, that group has reported on the 5 or even 10-year experience of 30 plus patients implanted with DBS, showing fairly impressive sustained responses. So some of what's going on in the research right now is continuing evolution to understand how to improve this, where to put the electrode, where to target. So on the right is just showing you one of the things they're focused on is really trying to find a sweet spot where maybe multiple white matter pathways, such as uncinate fasciculus, the cingulum bundle, forceps minor, may interact really in this subgenual space. And it's really about disrupting, putting the electrode in, disrupting maladaptive signaling kind of at this crossroads. That's kind of one of the main theories. So that's active work going on now. But watch this area for developments. For VNS, again, we don't have that much time to get into it, but there is a large randomized controlled trial. We're one of the sites for it going on right now to, I would say, reinvestigate VNS for depression. So as you probably know, VNS was approved many years ago, cleared by the FDA for treatment-resistant depression. This is a surgical procedure where a stimulator is implanted on the vagus nerve. But it hasn't been widely used. It's not covered by most insurances. So there's actually active collaboration between the device manufacturer and Medicare to conduct a large randomized trial to provide more information on its efficacy. This paper by Aronson from a few years ago, I think caught people's attention and was part of I think the reason for the current trial. And this is reporting on an observational 5-year follow-up on individuals with VNS versus treatment as usual. And you can see you get sort of a slow but cumulative response. And by the time you get to 5 years, you have fairly impressive response rates in the VNS arm compared to treatment as usual. There are methodological limitations. This is not a randomized controlled trial, but it is interesting. We've been talking about rapidly acting treatments. VNS seems to be a little different, sort of a slow accumulation of benefit, hopefully for individuals with really some of the most severe forms of depression. On the right, I thought this was interesting. The investigators just broke up the VNS and the treatment as usual group into those that had a history of response to ECT or not. Not surprisingly, those with, both arms, those with history of non-response to ECT did worse. But interestingly, if you look at for example, orange with the orange square on the right, those are VNS with a history of ECT non-response. They're still getting up to 60%. And these are sort of some of the most sick patients. So again, I think interesting. I think we'll know more in probably in a matter of months what the status is of VNS for TRD. A TMS, just a word about it. This is widely used now in the community. I think it's an important augmentation option for patients because it's not a medication and it's relatively easily accessible. Typical response rates in trials are about 40%, could be 50%, depends on where you look, with reasonable effect sizes. The classic standard approved dosing, as you know, is to the left DLPFC, 20 to 30 sessions of high stimulation, typically over 4 to 6 weeks. More recently, the intermittent theta burst protocol was cleared by the FDA. The main difference here is it's still typically given daily, just it's much quicker, 5 or 10 minutes versus 20 or 30 minutes for the standard. There's been a report on a modified sort of high dose functional MRI guided form of the ITBS from the Stanford group that I wanted to share with you because it's generating a lot of excitement. This was a small study, 14 versus 15. This was published last year in the American Journal, but this is an optimized form of TMS where individuals are getting multiple, they get 10 sessions a day of the theta burst, over 5 days. And you see on the lower right, the efficacy. It's very impressive, very large difference between sham. And they only get it for 5 days, and then you see the weeks out that response seems to maintain. Obviously, we'll have to see if this replicates in larger studies. One of the key features here is that they used functional MRI to detect the area of the DLPFC to target for each patient, actually based on its association with the subgenual. So again, it kind of gets at so this is a functional brain guided approach, which is kind of what we want in psychiatry, right? So this is another exciting development. So those were all the slides, and let me just wrap up for you. And I believe I've left a little bit of time for questions, which was my goal. So depression is a major cause of disability worldwide, and our treatments aren't good enough for a significant minority of patients. So just to wrap up, what I've tried to convey in a short amount of time is that new treatments targeting glutamate and GABA probably hold significant promise for the treatment of depression. We've already seen some come online, and we think hopefully there will be more. Inflammation is important, but the efficacy of anti-inflammatory approaches is sort of uncertain at this time. We've talked about a role for selective kappa opioid targeting. I've shared a little bit of our own data, early data on targeting the KCNQ channel for depression. We touched briefly on emerging data on the use of psychedelics and some advancements in neurostimulation. I hope that was helpful. I wanted to cover a lot, but I hope I didn't go too fast. This is the Depression Center. It's a huge team effort where we do clinical trials and specialized treatment. And thank everyone for your attention. I'd be happy to take questions. Now, it's not obvious to me where to see questions. Should I stop sharing? Let's see. Okay, let's see. I see Q&A. Okay, question about, you mentioned glutamate circuit when talking about ketamine. Is it only neurons or other cell types? That's right. Okay, so a question about glutamate system and yeah, you know, and again, obviously, I just wanted to really touch on and try to give you guys some references because it's a large topic. There's a question of whether astrocytes are involved, and I think the answer is yes. And in fact, there's a whole really interesting movement on targeting the role of non-neuronal cells in depression, cells like astrocytes, microglia. Both astrocytes and microglia are important aspects of regulation of glutamate signaling. So there is a way to bring in glial cells and astrocytes into the story of glutamate regulation. Okay, a question about DBS. Is the DBS appropriate for unipolar patients? Yeah, so a question about DBS and unipolar. So I should have been more clear in my discussion. Almost everything I showed you guys was focused on unipolar major depressive disorder. I talked not at all about bipolar depression. Apologies to everyone out there about that, because it's a huge topic. DBS has been primarily studied in the context of depression in unipolar. That's typically one of the requirements, at least for studies I know that Helen's group does. Not to say there aren't other studies in bipolar, but typically it's studied, and it's the same for TMS as well, that these are primarily unipolar patients we're talking about. Question about neurotrophins as a therapy. Yeah, that's a fair point. I mean, so a question about, if I understand it right, can neurotrophins be used as a therapy? In principle, yes. You know, again, you could look at this and what I presented and say, well, look, anything that can increase the availability of BDNF, say brain-derived neurotrophic factor, you know, could be antidepressant. I think a lot of the problems become what can get absorbed, what can you get into blood and into brain is not simple. In general, peptides are hard to get into the brain. It's probably a longer discussion, and I don't have all the answers, but it's certainly been the case that sort of straightforward things you would think, like increased neurotrophins, just in and of themselves are not that effective. Yet it seems that like almost every effective antidepressant we can find does increase neurotrophins. So I think jury's still out on that. Okay, let's see. Another question about targeting BDNF. Yeah, so targeting BDNF track B, I think that's, again, that's sort of a unifying theme that a lot of studies and a lot of folks have been working on, and particularly in basic science drug discovery. What are novel ways to augment BDNF track B signaling? I will say, it's probably important, I'm glad you brought that up. It's important to say it actually, and this might complicate systemic treatment, it seems to be pretty brain specific. So for example, a lot of the schematics I showed you, the reviews by Duman, that typically is showing the role of neuroplasticity being a protective, a positive thing in regions like hippocampus and prefrontal cortex. Conversely, there's been a lot of work, Eric Nestler's lab, many others, Scott Russo, many others, actually pro-BDNF signaling in more ventral and limbic regions, and in regions like the nucleus accumbens and the amygdala are actually pro-depressant. So actually driving neuroplasticity in those areas is bad. So that's an important like caveat to sort of the temptation, and what I've been sort of saying, you know, things that are, you know, increased neuroplasticity is going to be therapeutic. The caveat is only in certain brain systems and regions. So that's the issue. Comment on the recent scan. Yeah, I had a question about a head-to-head ECT versus ketamine. it's actually on my list. I need to look closer at that study. We're part of a large study funded by PCORI, wrapping up 5+ years. That's a similar, that's IV ketamine versus ECT. So it's very top of my mind. But I don't have any comments right now. This is a question about, there's a paper in one of the neuropsychopharm journals about ECT versus ketamine. But stay tuned. There will be a larger study coming out hopefully soon about that. Let's see, question about, S-methadone. Yeah, I saw that. I don't recall the details of that question about S-methadone study by Fava and AJP. I have to check that one out. Yeah, there's a lot of work in targeting opioid system. And actually, I in the interest of time, I really just focused on the Kappa because it's actually quite a bigger field. Obviously, we have to be very careful going down that road with targeting opioid signaling. Let's see. Okay, you're welcome. Question by Joshi. Okay. Well, hey, you guys did great timing. We have 1 minute. Question about nutrition. Yeah. Again, it could have been a whole talk, right? It's one of those things. There's just a dearth of good randomized controlled evidence. We know nutrition is important. We know exercise is important, but we don't have a lot of randomized controlled trials, and the whole nutraceuticals, nutrition, and herbal supplements. Part of the problem is it's outside of FDA regulations, so it doesn't. We just don't have a lot of data on it. But hey, Alex, thanks for joining. Yeah, so there was a great review actually by the Fava group, I think a few years ago now, in the American Journal, that was a very nice, thorough, scholarly review of all the data that we do know about. I think the topic was sort of natural and complementary and alternative treatments for depression, which is a really important, exciting area all its own. You're welcome. Well, listen, thank you everyone for your attention. This was a great opportunity. I hope you got something out of it, and look us up whenever you're in New York. All right, take care. Bye, everyone.